Seeing is believing. Many fundamental questions in medicine and biology can only be solved if we have the capability to directly watch what happens inside cells and tissues.
One of the cutting-edge technologies to visualize these processes is light sheet microscopy. Here, a thin sheet of light is used to quickly scan through a sample and create a three-dimensional image. This fast, gentle imaging makes it ideal for watching living cells and tissues and to observe how they grow, interact, and move.
Our group at the University of Rochester is developing a new approach to light sheet microscopy. And we can do that thanks to the generous support from the Arnold and Mabel Beckman Foundation. Our approach overcomes current limitation in the technology and will make it much more user-friendly. We brought together researchers from 4 different departments: biology, biomedical engineering, data science, and optics. And they collaborate in 3 teams. First, the engineering team that built the instrument is fine-tuning it. Second, the data science team that takes the images once they are acquired and cleans them up and does the image analysis. And finally, the biology team that uses this new technology to make novel discoveries in biology and medicine.
Light sheet microscopes typically use multiple objectives, one to shine laser light onto the sample and one to capture the image. This design requires a cumbersome setup to hold the sample in place in the small gap between these objectives, which isn't always compatible with the samples of a typical user. Our design employs a carefully aligned off-axis optical path to deliver the sheet of light through a single objective and then capture the image with the same objective. The sheet of light illuminates the sample at an angle and is then rapidly swept through the sample using mirrors underneath the objective. This design does not require a special mount to keep the sample in place, and therefore it can be used with normal microscope slides, multi-well plates, and other traditional sample holders, making it simpler to use. This flexibility allows us to stimulate our samples with drugs or nutrients while imaging so that we can directly watch how the cells react. Our instrument replicates the same ease of use that has made confocal microscopy so popular for biological imaging, but combines that with the gentle 3D imaging that only a light sheet microscope can achieve.
Light sheet microscopy is powerful, however, it also has its own inherent limitations. One of these is a spatially varying focus that leads to images that are more blurred in some parts of the scene than others. We address this issue by modeling the degradations in the imaging process, and then by feeding the observed data along with that mathematical model into a physics-informed AI method for reconstructing the underlying imagery. The resulting images are crisp, even in parts of the scene that would otherwise be unreliable. This, in turn, enables biologists to extract insights from challenging data.
My lab works with fairly large 3D bioprinted biological samples that are too big to image with typical microscopes. These prints contain bacteria that grow and divide over extended periods of time, which is challenging to follow by conventional imaging as well. We are using this approach to study a new idea for producing sustainable hydrogen fuel. It involves 3D bioprinting bacteria and incubating them with quantum dots. Imaging these samples with the light sheet system will let us understand how the bacteria interact with the quantum dots, leading to further advances in sustainable energy production in the future.
My lab uses small microfluidic devices to mimic human tissues that model healthy physiology and disease. One of the systems we model is the vascular wall during inflammation, when immune cells called leukocytes crawl out of the blood to reach the underlying tissue. To capture this migration, we need to image the immune cells both above and below the vascular wall. Light sheet microscopy is ideal for this, and our new system brings much higher resolution in the direction of migration. This will provide more information on how immune cells migrate during inflammation and drive our research in new directions.
To further improve our microscope, we are now including novel optical components into our instrument. Those components leverages Rochester's own revolutionary expertise in freeform optical design. In our original design, a pair of secondary objectives are used to rotate the skewed illumination plane onto the camera. This design sacrifices some resolution and requires a precise alignment that can be affected by small temperature variations. We can solve these problems with freeform optics, which offer a much wider range of design space solutions and can potentially achieve high resolutions, increased thermal stability, and better optical efficiency. We are really fired up to implement this next improvement in our microscope and to push the envelope of light sheet microscopy even further.